So Mendel performed the same test with all the different traits for the pea plant. He used purebred homozygous traits for each, or homozygous alleles for each trait. So he crossed the purple with the Y axial. You do not have to know each test that he performed, but you do have to know that every time you cross a purebred, you're going to get an F1 generation, which is also called a hybrid, and all the hybrids will be heterozygous for the trait. He later performed another test. He crossed the F1s, which basically were the offspring of the first cross. So he crossed a hetero and a hetero, and then he ended up with this. 3 of the 4 plants had purple flowers, and 1 of the 4 plants had white flowers. After this cross, he developed some principles. So prior to the cross, he had some ideas that led to these principles. Mendel said, if there are alternative forms of genes, they're called alleles. So example, a gene for flower color purple is one form, and a gene for flower color white is another form. Each organism has 2 pairs of alleles for each gene factor. You're going to get one from your mother and one from your dad. If the 2 alleles are the same, we call them homozygous. If the 2 alleles are different, we call them heterozygous. When one allele in a heterozygous organism is expressed or is visible, it's what you see, that allele is dominant. The one that does not appear is recessive. And whenever 2 alleles segregate, which means separate during the formation of gametes, so when a cell is producing a gamete cell, the alleles segregate. This is called Mendel's principle of segregation, one of the 3 principles. So he has the principle of dominance, principle of segregation, and principle of independent assortment. So Mendel's principle of dominance state that a dominant gene will always mask the recessive, as we've already said. So let's give you a scenario. Round is dominant to wrinkled. We're always going to choose the first letter of the dominant trait, which is R. So if I give you capital R, we know the organism's phenotype is going to be round. Capital R, little r, you're still going to have round because according to the principle of dominance, the dominant allele masks the recessive. Little r, you'll have wrinkled. So here's another example of Mendel's purebred cross with visuals. So round, we know is going to be capital R because it's homozygous or purebred. And wrinkled will be little r. If I cross these 2 parents, I'm going to get a round offspring because the dominant always masks the recessive of a heterozygous offspring. This cross is called purebred or homozygous cross, and the offspring is called heterozygous, or the offspring is heterozygous, and we call this offspring F1 or hybrids. So here's another scenario. If I tell you tall is dominant to short, we're going to use T because T is the letter that expresses the dominant trait. 2 capital T's, the organism's features will be tall. A heterozygous organism, genotype, capital T, little t, the organism will still be tall because dominant masks the recessive. And little t will be short. If I take these 2 and I cross them, capital T, and little t, all my offspring will be heterozygous. Same scenario for a different trait. Now, it's important to understand that everything you learn in genetics is based on chance. It's a probability. So whatever predictions you make are simply that. There's always going to be a 1 in something chance that the offspring produce will have the genetic trait. That's for each time of conception. So let's go over how to solve a Punnett square so you can determine what the probabilities are. I'm always going to give you a scenario. In this example, I'm going to give you purple flowers are dominant to white flowers, and I'm going to cross a hetero purple with a white. The first thing you have to ask yourself is what allele I'm going to use. And since I know purple is dominant to white, we're going to use the letter P because it's the first letter of the dominant trait. So for dominant, we're going to use capital P, and for recessive, we're going to use lowercase p. Next, we have to figure out what are the genotypes of parents. If I'm going to cross a hetero purple, that means I'm going to cross a genetic capital P, little p, or the genotype would be that, and the homozygous white, because in order to be white, you have to be homozygous recessive. So setting up the Punnett square, here's my first parent, here's my second parent. It doesn't matter where you put the parents. I generally choose the top of the Punnett square for the first parent and the side for the second, just to stay with some kind of consistent parent. So basically, you're going to drop this P and move this P across, and you're going to get capital P, little p. Again, drop this P, bring this P across, two little p's, drop, bring across, capital lower, drop, bring across, lower, lower. I'm going to come over here and open up that same Punnett square. Now, I can ask you several questions on the test. The first set of questions could be determine the genotype possibilities. Remember that these are possibilities. That means every time I conceive, there's a possibility that my offspring can be this or this. So I have half chance that my offspring genotype will be capital P, little p, or heterozygous. Half that my offspring's genotype can be little p. For phenotype, half will be purple. That would be this half right here. And half will be white. I'm going to use somewhat of a white color, which is right here, or lighter color. Let's do one more. Same scenario. Dominant is purple, but I'm going to give you new parents. This time I'm going to give you a homozygous purple parent and a heterozygous purple. So homozygous parent is the first choice. If they're purple, they're going to have two capital P's. Heterozygous purple, capital P, little p. Still purple because dominant always masks the recessive. Bring the P down, bring across, you got two capitals. Bring down, bring across, two capitals. Bring down, bring across, one capital, one lowercase. Bring down, bring across, one capital, one lowercase. So you've got genotype of parents, because that wasn't given to you. Capital P, little p. You saw that right here. And capital P, capital P, capital P, lowercase p. The genotype of offspring, half of my offspring are going to be capital P, which is right here. The other half is going to be uppercase, lowercase, which is right here. That means there's 50% chance every time of conception that you're going to have a genotype of capital P, little p. The phenotype in this scenario, all organisms will be purple, because if you're homozygous for the trait, you're going to definitely be purple. And according to the principle of dominance, the dominant gene always masks the recessive.